Good morning students, welcome to the next lecture of biomolecular spectroscopy, and in this lecture we were discussing the application of a spectroscopy in biopharma setting. And so in last lecture I gave you example where we will be using UV visible, where are we going to use CD, where fluorescence, where FTIR and Raman, and we also discussed a case study for that. Right. So, and I discussed you about biosimilars, right. So, biosimilars, just to bring you again on the same platform. Why biosimilars? The biosimilars, because they are cheaper and cost effective. So, you see there is a new drug that is approved and it is a biological drug. So, biological drug basically are revolutionizing the treatment of many complicated diseases such as autoimmune disorder, auto-inflammatory disorder, cancers and so and so for cardiovascular disease and all those. But when these diseases are discovered and marketed by a company, the cost is very high, right, and they also patent it because they need to recover the cost that they have put in the investment. So, those are called reference listed drug or new drug. Biosimilars are actually, once the patent is going to expire, many company can make these drugs, right, by just not infringing the patent, but once the patent is expired they can launch the same drug with much cheaper price, and that can be available to large mass. So, biosimilars are like then it becomes inexpensive, not so expensive, and you see. So, biosimilars drugs for say rheumatoid arthritis $50,000 per year, it is too high for a new drug, but biosimilar comes, then it becomes much cheaper, and like that is what cost for say TNF alpha I drugs increase since its introduction. So, cost estimate saving that can happen in next decade because we are going towards the biosimilar, and money can be saved in biosimilar, right. So, all those you be aware that biologics are protein peptide monoclonal antibody based drug, and that you have to go from a known DNA sequence. So, any virus, therapeutic serum, toxin, antitoxin, analogues, all of those which are used for treatment, prevention and cure of disease are can be classified as a biologics drug, coming from biological source, virus, serum, antitoxin, toxins, all those will be called biologics drug. They are derived from living organism, right. Living organism means they are cultured in bacteria or yeast or human cell, right, and like human or animal source or even plant source, many of these can come from there or some organism. These are therapeutics protein, and but to like they originate from here, but to do it using recombinant DNA technology you can do in lab scale, then you have to have a unique manufacturing facility, something like this. This is the manufacturing facility for biologics, and at the end you get a unique biologics, that is a therapeutic protein, right. So, a biosimilar is a copy of that commercially available biopharmaceutical, that reference listed drug or reference product, that is no longer protected by patent. So, this biosimilar drug is just a copy of the commercially available drug, but it undergoes rigorous analytical and clinical assessment to prove in totality that reference product and biosimilars are similar, and that has been approved by regulatory agency according to a specific pathway of biosimilar evaluation, and this is highly similar to the reference product in all terms, physiochemical, efficacy, safety, immunogenicity, all those. So, if this is highly similar to the reference listed drug, this will be called biosimilar, right. How do you establish the biosimilarity? We establish, so this is say a drug discovery pathway for an innovator molecule, new molecule. They do some clinical study, analytical study, then quite a bit of preclinical, then clinical pharmacology, and a huge clinical study that goes to bring a new drug. And therefore, if the clinical study is quite extensive, then the money involved in this will be huge. It can be very, very costly, and therefore, lots of these biological drugs are quite costly, right. It is the number many times is unaffordable by a common people from India. But if you are making a biosimilar, right. So, here you do not need to do that much clinical study, right. So, only conducted on sensitive patient population with sensitive endpoint and designed to detect difference if there is between the innovator molecule and biosimilar molecule. So, what you need to do is analytical, a robust many analytical techniques has to be invoked to do a comprehensive analysis of biosimilar. Then you do less preclinical and clinical, and finally, the end point is only small. So, if you look at this, this is the quite huge for biosimilar, and that is what in this course we were emphasizing, that the biophysical methods which comes into analytical portray has to be done robustly. And in the case study that I showed you in the last technique, how we are going to each of these using, that is extremely important, right. So, analytics, the physiochemical, physical or biological assay are basically eyes and nose and ear of any biomolecule, that sense basically. These are like as we can sense using our sensory organ. Similarly, analytical tools can sense the physiochemical biological assay. So, physiochemical, what I mean by primary structure of a protein, secondary structure, tertiary structure of a protein, all those techniques that I discussed with you, UV visible, CD, FTIR, Raman, all those can do secondary structure, some tertiary structure. NMR, X-ray, cryo-EM can do the tertiary structure. So, all of these analytical tools gives you idea about the physiochemical property or biological assay, physiochemical property of these comes analytical. Then suppose you are designing a drug. So, this drug has to bind to a receptor, and then it activates some pathway, right. So, that is coming under biological assay. You have to say immobilize on a chip like SPR chip and then pass the ligand and look at sensogram how it is changing. So, those assay can be called biological assay, or basically here the luciferase assay and many such assay, ELISA based assay are biological assay. So, that is what you have to do in this analytical assessment that I was talking to. So, physiochemical like means biophysical techniques you have to do, then one has to do the biological assay to establish this. So, range of analytics that is needed, something that we discussed and something was beyond of this course. So, microscopic based, mass spectroscopy based, you want to know the like a primary sequence of those proteins. So, you have to invoke mass spectroscopy based. Then flow cytometry, what happens to that on a cell you can do with proteomics, genomics, transcriptomics, gliomics, immunomics, what kind of immune markers are going, and then bioanalytical binding assay you have to do ELISA based, SPR based, MSD based techniques one has to invoke to use this range of like analytics has to be invoked for understanding the biological assay. Then cell based assay one can do signal transduction or transcriptional activation, and finally, you can go to preclinical study where you have a transgenic mice, small or large animal dose model, and actually at the end epidemiology. So, that is what we do, physiochemical and functional characterization of novel biosimilar we do. Primary structure you do by intact mass, right. Intact mass means like a whole sequence, then reduced mass by reducing disulfide bond, peptide finger meeting, disulfide mapping, element degradation to know the sequence of amino acids. Then you can do the glycoform analysis like glycan profile, one can do exoglycoside assay, monosaccharide, effucosylation, silation. So, all these glycoform analysis, especially in MAB, this analysis is extremely important. Then you do higher order structural analysis, far UV, near UV already we discussed, FTIR, intrinsic extrinsic fluorescence, and for stability you do differential scanning calorimetry, for 3D structure you do X-ray or NMR or multi-angle light scattering. So, all of these we have not discussed. The XDX-MS, native MS, nanoparticle imaging we have not discussed, but these upper one we have discussed. For purity you have to do size exclusion chromatography, charge variant analysis, reverse phase, the hydrophobic interaction and glycosylation, oxidation, deamidation, all of these you do. Then biological assay you can do effector function, like a receptor based study you have to do, and finally all of those data that comes you do data analysis interpretation to have a comprehensive characterization to establish the structural integrity for proving the totality of evidence for a drug approval, and that what requires here in the complete pathway that we are talking about for a biosimilar approval. Right. So, these are some of these structures. So, you do everyday kind of flow or imaging cytometry, laser scanning, and one point you do crystallography, electron microscopy, ESCM and confocal, all of these. If needed you can do it, right. So, what I want to emphasize for biosimilar or novel biologics, main thing is you have to show that if there is a discrepancy, why this discrepancy is happening. If it is not discrepancy, it has to give the totality of evidence that we talked. So, using all these a suite of analytical technique, we have to prove the totality of evidence that our biosimilar is same to the approved drug, right. So, all these you have to do. So, this has been like many of the clinical where there is a difference comes, right. So, the PK modeling can predict the faster clearance risk. So, not only analytical, some bit of you have to do also the animal model, anti-drug immunogenicity should not happen, and there should not be, even in PK it should not have been difference, right. So, it is not enough that UV visible, far UV or a SEC or anything can catch. Each technique answer different question, and therefore, you have to combine all of these technique, right. Just what we have studied in this course, UV visible can be done just to measure the concentration, but what it was not detected, the structure, right. Fluorescence it gives you tertiary instability, the residue level structure you do not know, dynamics you can get some, but not everything, right. So, now IR you can get a packing differences, CD you can get early unfolding, Raman you can get solid state or aggregation rates, but each of these technique is positioned uniquely to give you some answer, not all answer that is needed. So, that is actually important point to be understood, that we have to use variety of technique that are there to be combined for complete assessment, right. So, let us take the case study 2, how orthogonal techniques helps in detecting this discrepancy which would have been missed if only one or two techniques were used. So, now we take the monoclonal antibody. As you know, these are the prominent class of drug, it is a huge molecular weight. So, if you remember the structure, the monoclonal antibody here, it is a Fab region, FC region 150 kilodalton, 2 chain connected by disulfide, then glycosylation, lots of glycan variation, many things are there in IgG. So, too complicated even to make biosimilar, even if you know many things, to get this biosimilar right, it takes years of research and extensive data generation. So, now how we can take this as a case study to understand how orthogonal technique can help in detecting the discrepancy which would have been missed by one or two technique. So, biosimilar that we are taking is IgG MAB. It has a hidden tertiary structure discrepancy that can be only detected with orthogonal technique. So, the product that we are taking is IgG monoclonal antibody biosimilar. Now, development stage, right, if we are doing late analytical compatibility or pre-PK, we are invoking this analysis, and manufacture has been done in CHO cell, Chinese hamster ovary cell. Now, then we purify using protein A capture, and cation anion exchange polishing has given us pure protein. Now, what we have to do? initial conclusion highly similar to the reference product, but maybe it is incorrect. Let us look at how and what. So, we are starting with extremely simple experiment of UV visible absorption. Remember, this is the most easiest technique that one can do, it is a cheaper one. What you are measuring? The absorption at 280 giving concentration, and then a spectral baseline we are doing 240 to 340. So, concentration difference between biosimilar and your novel biologics, concentration difference is less than 1.5%. No light scattering or turbidity is happening, and spectra indistinguishable from the reference product, right. So, that's we are getting in UV visible. So, what it suggests? No discrepancy detected, material are equivalent, that is what we are saying here. Now, step 2 we are doing far UV visible CD experiment that we are carrying out secondary structure between this 190 to 250, and we can also do using the far UV CD thermal unfolding. So, this gives overlapping beta set structure dominant and indicates that melting temperature are more or less similar. So, here again no discrepancy detected, it suggests that the secondary structure in monoclonal antibody appears to be quite similar. Now, so UV visible gives us similar, CD is giving also similar. Now, next we move to third experiment, which is near UV CD, gives tertiary structure information, and what we are measuring is aromatic amino acid between 250 to 320. Small but reproducible reduction of tryptophan intensity at 295 we see, slight distortion that was happening in tyrosine at 275, effect seen in multiple biosimilar lots, but we can also say it could be noise, but here that was noticeable. So, it is not a random noise. Let us see how it is. So, near UV CD we know it is a tertiary, it reports about tertiary packing, and changes indicated altered microenvironment despite of identical secondary structure. So, see we are detecting something is changing here between biosimilar lot and RLD. So, what it says discrepancy we can detect it, the fold is globally correct, but packing seems to be different as we move from secondary to tertiary structure. To strengthen that we move to intrinsic fluorescence. So, what we are measuring here is a tryptophan fluorescence, right, emission that exciting we are doing at 295 and measuring emission between 320 to 340, and we can also, if you want, we can stress little bit sample and agitate it and do the fluorescence experiment. So, what we found even in the lot to lot variation, that there is a red shift happening of 3 to 4 nanometer compared to reference product, and that gives faster intensity loss under stress. So, this means this technique is detecting essentially if we are stressing, intensity is lost, getting loose, but here in the biosimilar lot and reference lot there is no stress, just we are measuring the sample, but even in that sample 3 to 4 degree of shift is happening. So, what it suggests that tryptophan residue are more solvent exposed, compared to the reference listed duct. So, it indicates like in the stress sample the loss of some tertiary structure you are doing. CD could not alone detect it, but now we are kind of conforming. So, we are conforming the discrepancy protein although looking folded, but it is less conformationally stable. That is what one can detect, that biosimilar whatever a manufacturer is manufacturing, overall it looks fine, but it is not that stable, right. So, next we move to the next technique that we had discussed in this course, FTIR. Here we read amide 1 region in a say lyophilized one. So, if it is not stable, we can check it whether it is lyophilization keeps it stable, or humidity stress we give 25% to 75%, and what we found that slight increase in intermolecular beta sheet signal is happening, and reference product did not show any change. Even if you put it humidity condition, RLD is not changing, but your biosimilar is changing. So, that says that increased aggregation propensity is happening in biosimilar in solid state, and that could link the altered tertiary structure package. So, now we can even identify the source of discrepancy. The problem persists in the final dosage form, that is in the solid. Now, if that means one can really avoid this discrepancy if we do it correct at the beginning stage. Now, to confirm that, we again go to Raman spectroscopy. We perform the Raman spectrum of formulated dose and compare across the multiple variants. What we found that subtle change that is happening in aromatic ring mode, disulfide associated, and this differences can be tracked back to the late stage polishing stage, where we are purifying the protein. Probably there something has changed. So, it is important that slightly difference in redox microenvironment during the polishing is changing this structure. So, can we go back and correct it, right. So, the altered disulfide scrambling is happening, primary sequence unchanged, but folding pathway seems to be different, and that difference may be coming during purification state. So, now it is job of a scientist to go and fix it. So, root cause of discrepancy, what we found that Raman can identify the structural discrepancy to the process, but not random variability. So, if there is a structural discrepancy that is coming out from the purification state, if you remember what we are doing here, just to put some bit of like here we are purifying the protein somewhere here. So, after this production we did, right, and after the production we were going and doing the purification of that protein using various column and all those, and then we could detect here that something is changing in this case, and if that change, if this is changing here, it is going to impact here. So, that is the problem one can face it if the earlier stage we could not face it if earlier stage analytics is invoked in this study. Okay. So, what is the key takeaway? That no shortcut in biosimilar production, and therefore, FDA insists on orthogonality. You cannot rely on one technique, you have to use many techniques which are orthogonal. FDA reviewers knows that clinical efficacy in trial are insensitive to the subtle change that happens at molecular difference. So, orthogonal analytical technique can detect the problem that may happen at very early stage, and more precisely even if a side chain is moving here and there, if your methyl, suppose methyl is rotated by this, you can detect using any of these orthogonal technique. That may not have too much clinical efficiency, but you can look at a structure and find it out your methyl side chain is rotating or more flexible, and that could have some implication in aggregation and long term impact. So, this is the case why FDA now says comparative analytical assessment is generally more sensitive than a clinical study for detecting meaningful difference. So, new guidelines that are emerging from FDA insisting on a robust, comprehensive, comparative analytical assessment, right. So, why this matters under the new FDA guidelines? The guideline says, right, you can have a new amendment in the guidelines. It says that analytics can replace clinical study only if residual uncertainty is low, right. So, that means analytics you have to really do it very thoroughly. Orthogonal discrepancies can reintroduce uncertainty. So, when uncertainty reappears, clinical data is required again, but if does not, if you have done this very rightly, very robustly, thoroughly explain all data in a robust manner, probably clinical like a clinical efficacy test may not be that robustly allowed, and that is what one can say it in biosimilar production. Actually, you do not need to go too much into the details. of the clinical trial. You can essentially employ the variety of technique that I discussed here, like you do start with a primary structure, you do intact mass, reduced mass, peptide fingerprinting, disulfide mapping and sequencing, all of these. You see this you required a very expert team here, right, to work in this. Then comes the complexity in my glycoform analysis has to be done again robustly. Here you also requires a very expert, the mass spec operator, the data analytics and all those. Tons of data is emerging, and now that data analysis and interpretation becomes very important. In this course, what we are discussing these techniques, the higher order structure, again each of these techniques are involved in themselves. I only discuss up to fluorescence and Raman, but think about DSC gives thermal stability, X-ray, NMR, multi-angle light scattering, all of these are again extremely involved technique and you require expertise here. Then during purification you require a downstream processing guys who knows very well size exclusion chromatography, charge like a ion exchange chromatography, reverse phase. So, again you require a great deal of expertise. Finally, you have to have a bioassay lab where you can see the effectiveness in the cell based assay or receptor based assay. So, one can think of how many experts team is required for doing a biosimilarity analysis, and therefore, these companies requires the experts from many different fields, not only for data recording, also for data interpretation, data integration, right, so that you can make a comprehensive informed decision about the quality of product that is going to go into the production and finally formulation and then into market and finally into patient. So, the collective effort is needed to bring a biosimilar and lots of expertise, orthogonal expertise is needed, and therefore, this comprehensive characterization is required for getting approval of a biosimilar drug, right. So, I just want to emphasize that there are plenty of career opportunity if you are really looking for in biopharma product, right. So, each of these techniques requires one expert to be there, and you can think of that how many experts will be needed to bring out a biologics drug or biosimilar drug in future, and therefore, it has a great potential to open an avenue for you in the biopharma sector. So, with all those let us close it, and now in the next class we will go to the next topic. Thank you very much.